Hello, everyone, and welcome to BrainMap. This seminar series is co-sponsored by P41-funded Center for Mesoscale Mapping, hosted in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Kristopher Kahle. Kristopher Kahle graduated from the University of Chicago in 1999 with degrees in biology and philosophy. He received his MD from Yale School of Medicine in 2007, where he also received in his PhD working in the Laboratory of Human Genetics, geneticist Richard Lipton. He completed his neurosurgery residency training at MGH in 2014, as well as postdoc research fellowship at Harvard University with co-mentors Stephen Ellich and David Klempam. He completed a clinical pediatric neurosurgery fellowship at Boston Children's Hospital, where he was Shelley staff associate and instructor of neurosurgery at Harvard Medical School. Dr. Kahle returned to the New Haven to join the Yale School of Medicine faculty, where he was assistant professor of neurosurgery, pediatrics, and cellular molecular physiology, and director of neonatal and congenital and anomaly neurosurgery from 2016 to 2021. Dr. Kahle is currently the director of pediatric neurosurgery at MGH and the Harvard Center for Hydrocephalus and Neurodevelopmental Disorders. He's a board-certified pediatric neurosurgeon with experiences in broad range of pediatric neurosurgical disorders, including brain and spinal cord tumors and cysts, all type of hydrocephalus. Kahle carry malformation, spina bifida, vascular malformation, and others. I would like to remind our audience to address any questions using Q&A box or raise their hands at the end of the talk. Dr. Kristopher Kahle, thank you very much for coming today, and the stage is yours.
Thank you. Thank you for the kind introduction, and it's a pleasure to be here. As was mentioned, I'm the chief of pediatric neurosurgery over at Mass General, so a practicing pediatric neurosurgeon who has some patients this afternoon to see, but also a funded investigator with a lab on the 6th floor of this building, actually. Today, we'll be talking about brain ventricles as windows into neurodevelopment and disease, and perhaps not just brain ventricles, but also CSF spaces more generally. These are two MRIs of a patient with congenital hydrocephalus and a different patient with another CSF space, cerebrospinal fluid space disorder called arachnoid cysts. These are my disclosures and funding. I have no financial conflicts. The lab is funded by 3 primary R01s and some foundation funding as well. These are some examples of conditions treated by pediatric neurosurgeons and adult neurosurgeons as well, illustrating that some of the techniques we use are pretty medieval and archaic. On the upper left-hand corner, as an example, to treat a traumatic or ischemic cerebral edema, we take one half of the bone off on the side of the skull, called a decompressive hemicraniectomy, and that can be life-saving until the swelling comes down, and then we put the bone back on. The middle picture, upper middle, is a patient with hydrocephalus due to a choroid plexus papilloma and oversecretion of CSF into the ventricular space. And to treat that, we essentially impale the ventricular system with a catheter that's about the diameter of a straw and feed that catheter down to the patient's peritoneum, where it serves as a continuous conduit for CSF reabsorption. Upper right-hand corner is depicting a young patient with medically intractable epilepsy, not responsive to anti-epileptic medications. And now, while we do more functional hemispherectomies than anatomic hemispherectomies, this operation is still performed where we lop out major portions of the brain to eradicate seizure foci. These procedures can be life-saving, but I would contend that there can be increased precision with which we choose our treatments as we increase our understanding of the molecular pathophysiology of disease. So pediatric neurosurgical disorders, a lot of them are sporadic congenital structural brain and spine disorders. Many of them appear radiographically similar on MRI, but in fact behave in very clinically heterogeneous ways. Hydrocephalus is perhaps one of the prime examples where many cases of patients designated with quote unquote hydrocephalus are characterized by big brain ventricles by definition, but have radically different physiology, which has ramifications not only for the choice of treatment, but whether they should be treated at all. Interestingly, many of these structural brain disorders are also associated with other neurodevelopmental deficits or neurodevelopmental disorders, such as autism spectrum disorder, epilepsy. So you have structural brain disorders, but rarely in isolation. Then they're usually also coupled with other functional neurodevelopmental disorders, which may or may not respond to the surgical treatment that's addressing the anatomic abnormality. So it becomes very complex very, very quickly. Despite that, as I illustrated on the last slide, as neurosurgeons, we have a relatively limited tool set. Our repertoire is admittedly small. And so we employ what could be considered one-size-fits-all treatments to a battery of different pathologies. And then we're confused when there are variable clinical responses to treatment. Perhaps we shouldn't be. So one result of our poor understanding of disease is because we poorly understand the genetic etiologies, which has always been a direct window into disease pathophysiology. So this is just a slide to illustrate the complexity of any important biological system. This is a famous wiring diagram, one of the first real shots at coming up with a systems biology approach to understanding blood pressure regulation. This was devised by Dr. Guyton in a famous paper and then recapitulated in his famous textbook of medical physiology. And the point here is that any biological variable that's important has been evolutionarily defended by a series of overlapping redundant systems to protect that variable. So whether that variable is blood glucose level or blood pressure, there are a number of overlapping redundant systems whose regulatory pathways, its job is to maintain homeostasis. So the question here is that for whatever phenotype you're interested in, for example, hydrocephalus or cerebrospinal fluid dynamics, in this labyrinth of overlapping regulatory pathways, what are the absolute essential nodes or genes or amino acid residues within those proteins encoded by those genes that are critical, absolutely essential, the most important for system behavior as a whole? Because once you know that, those can be prime drug targets. They can be modulated by either genetic or pharmacologic approaches to influence system behavior in a major way. And so the question is within this complex network, what is your approach going to be to define those nodes? And I would contend that human genetic approaches in general, but when you're dealing with human diseases, human genetic approaches are the most direct, efficient way to gain an understanding about human physiology. It's an unbiased agnostic approach. Instead of just saying, okay, I'm most interested in this gene for whatever reason, and I'm going to immediately come out of the box targeting it with a hypothesis-driven approach, we take an agnostic, unbiased approach. Our only bias is saying that genetics is powerful, and this is the approach that we're going to take. And we examine the human model system, so-called, knowing that whatever we find, it begins and ends with human patients. It's certainly inspired by the patients that we treat. All the diseases that we study are diseases that are in my patients that I operate on in the operating room, that I see in clinic, that I interact with their parents. Discovering genes for these disorders not only provides insight into disease pathogenesis, but because of the types of diseases that we're focused on, they can also illuminate very foundational aspects of human brain development that we automatically know is relevant. In doing this work, we feel, and we have examples, that we engender a more precise diagnostic classification scheme. So currently, a lot of the classification schemes of our disease are anatomically based, imaging focused. And for some of the reasons that I mentioned earlier, despite radiographic similarity, there can be significant clinical and genetic heterogeneity. And we have been finding that the genetic heterogeneity can explain the clinical heterogeneity. And when you take a term like hydrocephalus, really this is a bit of a garbage term that can be broken down into multiple subclusters of disease defined and driven by genetic mutations that, with the knowledge of that, different treatments can be employed, prognosis can be given, and more truthful and productive conversations can be had with moms and dads who are about to have a child, with a condition or about to be taken to the operating room. And beyond that, identifying potential therapeutic targets that can transform some of these diseases, which are classically considered neurosurgical disorders, only really managed by neurosurgeons, into non-surgical diseases. So we really use three types of data, all generated from our human patients and their parents or other family members, which includes genomic data, right, DNA, and DNA made possible by next generation DNA sequencing, largely exome sequencing for a lot of our work, but now also whole genome sequencing. We use transcriptomics these days, usually single cell, single nuclei RNA sequencing derived from our own patients, as well as existing atlases that seem to be coming out almost weekly. And third, from the phenomic angle, and this is work that I think we've really pushed on, and work that I think many people and maybe in this room could, with even better expertise, contribute to, is really deeply phenotyping our patients from a neuroradiographic point of view, and also from a clinical history point of view. So we've been really using new artificial intelligence-based mining of the electronic medical record to make associations of patients' mutations with other under-recognized phenotypes that are starting to subclassify and subcluster disease, and linking radiographic patterns with genetic identity, seeing how similar some of these scans can be for specific subtypes that you wouldn't necessarily realize unless you knew their genetic identity and had their scans at the same time. By integrating these three data sets, you can not only define the DNA mutation that's causing the disorder in the patient, but then you can begin to gain insight for hypothesis-driven experiments of where that gene's protein product is expressed in both time and space, right? Allowing you to pick different Cree drivers for specific experiments. And then also on the phenomic side, making those links between genetic identity and MRI appearance. So anybody can look this up. It's kahlelab.com. I bring this up just because as an example of how we use the web and social media as just one other methodology, one other approach in our lab. So you can see join our studies. And that's, our outreach via social media has allowed us really to recruit patients not only across the country, but also internationally. I think some of the classic paradigms of how you set up some of these studies, where you pick a couple of different sites with maybe supporting physicians, and you set up infrastructure. This is extremely cumbersome and takes a very long time, and the yield is quite low. Whereas though it took a lot of time on the front end for all the IRB and HIC approval and NIH approval, what we're able to do now is really contact closed groups on Facebook where people for individual rare disorders are all gathered in one place, make them know that we're doing this study. Once they contact us, then that establishes a relationship in which we can have a dialogue and, for example, obtain consent, further talk about what the study's goals are, how they can get involved. And three, again, this is now works internationally, given some of the shipping methods that are available and also how DNA can be stored. But we can send out FedEx packages with our swab kits that people can do in the comfort of their own home. They don't need to go anywhere. You don't stick a kid with a needle. So it lowers the barrier for bringing someone into the study. And then they send it back in a self-enclosed envelope. And within a couple of weeks, we can define a gene mutation for a specific patient if the biology is ready to deliver itself to us. So the fundamental goal of the lab really is to translate omic discoveries into insights that improve the lives of children. Again, all this work is inspired by patients, uses human data, and then has a goal of using whatever insight we and others might find to actually improve the lives of patients. So there's two types of hydrocephalus. Today, I'm mainly going to talk about the primary form, also called congenital developmental, now realizing also genetic. And there's a whole other story, which I think is equally interesting on the acquired side. This is a type of hydrocephalus that arises due to some other known insult, secondary insults, such as infection like meningitis or brain hemorrhage from germinal matrix hemorrhage in a premature baby, for example, or a neoplasm, for example, a brain tumor squashing the aqueduct. And so these are just some examples of some papers that we've that depict our work centered on congenital hydrocephalus, and that's what I'm going to talk about today. Mainly because what I want to try to get across is that these some of the genetic models of hydrocephalus that we've set up in animals now, whether it's from mutating specific genes in populations of cell types that are important for hydrocephalus, given the integrative genomics that we've done, or using patient-specific mutations in these animals. These animals are great for doing imaging-based studies. And very straightforward, as opposed to some of the models of acquired hydrocephalus. These are a couple of examples of papers that have come out from our lab in the last few years on this form of hydrocephalus, which are a little bit more cumbersome, where a lot of them are surgical models. A lot of them have implanted hardware in their brains, which can confound the use of MRI based modalities to do some imaging studies. But these genetic forms of hydrocephalus really lend themselves to study via imaging based approaches. And a lot of the phenotypes are incredibly robust, highly penetrant, and just frankly fun to work with. So if you don't know what hydrocephalus is to begin with, and I don't blame you, despite the fact that it's one of the most common congenital brain disorders in the world. It affects about 1 in 1,000 live births, and it's really characterized by expansion of the cerebrospinal fluid filled brain ventricle spaces. This is a problem because in certain forms of hydrocephalus, that expansion of those fluid-filled spaces in the confines of the rigid cranium, by definition, and according to the Monroe-Kahle doctrine, which bears out in real time at the patient bedside, that increases intracranial pressure, intracerebral pressure, so-called ICPs, and because that can lead to devastating and lethal brain herniation if not addressed properly. The majority of cases of hydrocephalus, the congenital and developmental hydrocephalus, the cause is unknown, and has really been thought to be due to a balance of CSF production and CSF reabsorption, based on evidence and some data largely derived from acquired hydrocephalus cases. So those associated with hemorrhage, infection, and neoplasm, in which we know that there is either an increase in CSF production or there's some sort of lesion obstructing reabsorption of hydrocephalus, so that when you put in a shunt catheter into that CSF filled space, the ventricles can acutely be reduced in size. The patient can, within minutes, in fact, get better because the intracranial pressure is reduced. Whereas this other type of what we call hydrocephalus, because it's characterized by large brain ventricles that look exactly the same as the acquired cases, the physiology was really not as well known. And some other observations bear out that there was probably something else cooking in the pathophysiology of this disease, because we know that, in contrast to the acquired forms, when you put a shunt catheter into subgroups of these patients with congenital hydrocephalus, their ventricular size may not change, or their associated neurodevelopmental phenotypes do not get better. And so the question is, well, what's going on and why are the brain ventricles large? So again, focusing now on the left side of this, this little diagram in the primary congenital developmental forms. Obstacles to progress in understanding the disease for congenital hydrocephalus was really because no one really took a large swing at the genetic basis of this disorder. There were really only a couple of genes that had been discovered even long ago using traditional positional cloning approaches. The prototype here was mutations in the L1 gene causing something called L1 syndrome, which is really a neurodevelopmental disorder of which a significant number of patients have large brain ventricles due to aqueductal stenosis. The aqueduct is this fascinating passageway, aqueduct by definition, that connects the third brain ventricle to the fourth brain ventricle. And it's like the critical bottleneck through which a lot of CSF has to flow. And so if there's any stenosis or narrowing of that structure, you can get accumulation upstream. It's like building a dam in front of a river, so to speak. And so with the advent of exome sequencing that really started to gain popularity in 2008, 2009, and then has just skyrocketed, leading to the discovery of multiple disease genes, and particularly those sorts of mutations that had gone undiscovered using other traditional genetic approaches, because with exome sequencing, you can take an unbiased look at each gene in the genome. Of course, you're restricted to coding regions. That's the improvement that whole genome sequencing provides. But with whole exome sequencing and looking at the proband or the affected patient, and then mom and dad, you can not only identify familial factors like autosomal dominant and recessive contributors to disease, but perhaps more importantly, for some of these sporadic congenital brain and spine disorders, you can uncover pathogenic de novo mutations that occur after egg fertilization, so-called post-zygotic mutations. And so to uncover the genetic basis of congenital hydrocephalus, we set up something called the HydroSeq platform. The approach was really to set up an integrative genomics approach to discover, validate, and then begin to gain mechanistic insight into the genes and mutations that cause congenital hydrocephalus. So over the past few years, we've done this. It's a NIH-sponsored, very large consortium. We've been able to ascertain and phenotype the largest congenital hydrocephalus cohort in the world that now I think is a little bit over 2,600 trios. It's a family-centered social media engagement approach, kind of a grassroots approach that I talked about earlier. Despite that, we still, of course, have physician-directed referrals. I'm on staff at Boston Children's Hospital. We sequence a lot of their patients. We have great collaborations with Children's Hospital of Philadelphia and some other outstanding pediatric hospitals. Of course, we treat all our own. We try to recruit all our own patients that we take care of at Mass General Hospital. But it's really, in my mind, the social media approach that has made really big gains in a relatively limited amount of time. And so using exome sequencing, now whole genome sequencing, and then 500x deep sequencing in brain tissue derived from our own patients when we take care of them in the operating room, we're not only able to define inherited and de novo mutations in the germline, but also somatic mutations that are disease modifiers and further contributing to the patient's phenotype. After we define genes from a genetic approach, we validate and study high confidence genes that are of particular interest or novelty or potential drug targets using both frog and mouse. Frog is a bit of a medium throughput organism in which we are able to further probe the pathogenicity of the specific mutations for which genetic evidence, I would say, is isn't as robust, but still compelling. And then mouse and sometimes rat, if we're interested in probing the CSF dynamics of these things, of contributors of these genes, is really our organism that we use to look into disease mechanisms and physiology. This is Peter Jin's paper from our lab that was published in 2020. This is the second rendition or second version of our swing at congenital hydrocephalus using exome sequencing. The first was published in Neuron, this in NatMed in 2020, and hopefully the third will be even larger because it encompasses almost 6x number of individuals this summer. So that will be version 3. New genes, further increasing the depth of our knowledge of this disorder. But this was 381 probands, and we found that about 2.1% of these sporadic cases had mutations in genes that had been previously implicated, including multiple patients with L1CAM mutations. So these are genes that really you would think that if you had mutations in them, you would know, because the patient, given other phenotypes that they have, which are indicative of a syndrome, would be sent for genetic sequencing. But that really wasn't the case. There are still a bunch of people with mutations, even in known genes, that were not genetically defined. And then beyond that, about 18% of cases could be attributed to this mechanism of damaging de novo mutation. So a sporadic disorder, not inherited mutation, de novo mutation. At the cohort of this size, about 18% of patients, but now we're finding that about 23%, so nearly a quarter, 1 out of 4 patients has this mechanism of de novo mutation. So 12 genes had more than 1 protein damaging de novo mutation, and multiple genes had more than 2. So that's an extremely rare event in a cohort this size, a bit like getting struck by lightning a couple of times in the same day. One of these mutations, the most common of which was TRIM71, I'll focus on, along with P10, just because they're the 2 most common genes that continue to come up again and again and again. And of course, you guys recognize P10, probably are maybe less familiar with TRIM71, but this is data not yet published, in progress now, that will be coming out this summer, that shows the 2 most commonly mutated genes among a cohort of nearly 2,700 congenital hydrocephalus trios. So these are some of the de novo mutations that are in this molecule called TRIM71. It was a gene that was first discovered in C. elegans. It's important for neural stem cell fate transitions, actually stem cell fate transitions generally early in development. And its function is to bind to specific mRNAs and regulate their degradation, and in doing so, gene transcription generally. So it's interesting that these mutations in TRIM71 that we identified in congenital hydrocephalus tend to cluster in these so-called NHL binding domains. These are domains that are important for TRIM71's binding to its mRNA targets. And so we find that these mutations are disrupting these binding events. So of course, there's multiple mRNA targets. For some of these targets, it's a loss of function. Others, it's gain of function. So it's most likely a so-called neomorphic effect of these mutations that all cluster in specific residues. You can see the R608H and the R796H mutations both in the third position of their respective NHL binding domains. It's fascinating biology. Zui Fan, who was an MD PhD student in my lab, F30 funded, matched at UVA neurosurgery just a couple of weeks ago, and has a real deep interest in brain development. Went on to look at the mechanism of these mutations in a very nice model system. In fact, a few of him that he devised. So in contrast to our original hypothesis that in congenital hydrocephalus, we would be finding perhaps gain of function mutations in ion transport in water pathways in the choroid plexus. This is a tissue that secretes cerebrospinal fluid, or perhaps in other components of the CSF secretory reabsorption apparatus like cilia or the ependyma, other components that facilitate the transit of fluid through the system. That's not the story at all, and the biology is way more fascinating. And I think this really underscores the utility of an agnostic approach, a hypothesis-free approach, at least in the gene discovery phase, because this disorder is really all about neural stem cell fate, regulation early in development. The neural stem cells really reside in a ventricular zone, subventricular zone niche right under the CSF filled brain ventricles. And so I'll go into two examples of really a yin and yang example of how dysregulation of neural stem cell growth driven by genetically encoded mutation in the germline can cause big brain ventricles. So TRIM71 through the integrative genomics and single cell approach that we took, we really found that is a novel marker of neuroepithelial cells. This is the earliest neural stem cell population in the brain. And in contrast, it's not expressed in the choroid plexus, it's not in cilia, it's not in other parts of the brain, it's not expressed in the adult brain. So it's a very, very early marker of neuroepithelial stem cells. So TRIM71 patient mutations recapitulate hydrocephalus in mice. So if you make this single base pair change resulting in this amino acid substitution at 595, which is the orthologous residue in mice, to that, I believe the 608 mutation, you're able to recapitulate the massive ventriculomegaly that you see in human patients when the mutation is present in its heterozygote form, as it is in human patients. Homozygote mice with this point mutation end up developing exencephaly, a very severe phenotype, essentially lack of brain development, and then are embryonic lethal. So in addition to that, you can also recapitulate hydrocephalus by selectively or conditionally depleting TRIM71 in prenatal neural stem cells, whether you use EMX1 Cre or Nestin Cre. These are just two different neural stem cell specific Cres, in which if you deplete TRIM71, you also get a similarly huge brain ventricle phenotype. If you look at the both on the cellular and molecular pathophysiology in these mice, what you see very strikingly is an impaired prenatal neural stem cell regulation phenotype, in the sense that there is a precocious differentiation and decreased proliferation of neural stem cells in the ventricular zone. And what that does, of course, is it depletes the available neural precursors that are available to populate the developing cortical mantle. So I alluded to this a little bit earlier, but the recurrent TRIM71 mutations all localized to these NHL binding, these RNA binding NHL domains, and that I think we've now found and tested close to 17 mutations, all of which are clustering in these NHL binding domains that have very profound impact on their mRNA targets that are all very critical neural stem cell regulators. So the interesting part is that this thinned cortex that is in these congenital hydrocephalus mice and human patients is therefore really not a result of fluid buildup, right? It's not increased CSF reabsorption. It's not due to really decreased CSF, it's not due to increased CSF secretion. It's not due to decreased CSF reabsorption, but it's really impaired neurogenesis. So it's a bit of a Rorschach test when you look at something like this, which again, if it's an acquired hydrocephalus patient, if you don't treat that patient and catheterize that patient, sometimes within hours of presentation, they're going to die because of brain herniation and increased high ICPs, right? But in the setting of TRIM71 and multiple other congenital forms of hydrocephalus, this thin cortical mantle is just decreased neurogenesis. And so really it's the brain ventricles are just secondarily expanding, so-called almost ex vacuo dilatation, in which CSF is pooling, but it's really a primary deficit in brain parenchymal development. And so that's a bit of a shift in our thinking in the pathophysiology of the disease, which has huge implications for how we talk to patient families about the associated neurodevelopmental phenotypes of their patients, about what you can expect from a shunt. It really calls into question for subgroups of patients that have these mutations, whether they should be treated at all. And so it can also explain some of the varied responses to treatment that patients have when they're treated with some of these one-size-fits-all CSF shunting approaches. So as you would expect from a hypoplastic and very thinned cortex, in and of itself, it has very different mechanical properties that influence the interaction between CSF and the brain parenchyma at the CSF-brain interface, which probably lends to a secondary pooling of CSF and inefficient flow through the system because the space is just so big. And so that it's adding insult to injury, but not primarily a disease of either CSF secretion or CSF reabsorption. So this is just to put into words a very, I think, a fundamental point from this work with TRIM71 that's being recapitulated in several other, early neurodevelopmental genes, is that congenital hydrocephalus results from defective neurogenesis that leads to a hypoplastic, or what we call floppy, cerebral cortex that can facilitate secondary ventricular dilatation, ex vacuo dilatation, with preserved CSF circulatory functions. So if you, and we've done this in these mice, if you set up assays to measure, cilia motility or check the CSF secretion rate, these things are normal, despite the fact that there is a clear, huge CSF space. And so when you see and begin to define some of the genes that are causing congenital hydrocephalus, we are beginning to see that a lot of these genes are overlapping with other neurodevelopmental disorders, especially with autism spectrum disorder, some of the cancer overgrowth syndrome disorders, other developmental delay, intellectual disability disorders. SMARC-C1 is another one of our very commonly mutated genes. This is a member of the SWI/SNF chromatin modifying complex that I'm sure some of you know. And then multiple genes in the PI3 kinase signaling pathway. So genes you've all heard of, implicated previously in autism, other neurodevelopmental conditions, overgrowth syndromes, cancers. But the issue here is how that you can probably get the specific congenital hydrocephalus phenotype is that in addition to the germline mutation, there's probably some other second hit mutation in the gene pathway or in the specific gene that was implicated in the germline syndrome, or the germline mutation that gives the specific phenotype of interest for congenital hydrocephalus. And so this is really, or at least what I think is interesting. We've been modeling P10 and creating congenital hydrocephalus mice with P10 mutation, very similar to the approach we took with TRIM71, where we're able to selectively deplete P10 in the embryonic cell type in the brain in which it's most highly expressed by far, NKX 2.1 neural stem cell population, which goes on their descendants, of course, are GABAergic inhibitory neurons. So not the stem cell population that we would have thought of per se as the critical locus of how PTEN mutations are impacting congenital hydrocephalus, but that is the case. And whether it's selective depletion of PTEN in NKX 2.1 Cre driver lines or specific patient mutations, you still get the same huge brain ventricle hydrocephalus phenotype. The difference being, and it's the complete opposite of the TRIM71 story, is that in these mice, you get P10 deletion leads to increased mTOR signaling and hyperproliferation, not hypoproliferation, of this neural stem cell population. Because it's in the median ganglionic eminence that surrounds the paraventricular space around the aqueduct, what you do is you get brain overgrowth, aqueductal stenosis, upstream ventricular dilatation, and these mice are very dependent on CSF drainage, right? If you don't do anything, they die around P21, and because of the hyperactivation mTOR phenotype, we can correct the ventriculomegaly and also the associated cortical pathology, right, that leads to epilepsy and some of in kids a neurodevelopmental delay by giving postnatal rapamycin or everolimus, and on the genetic side, crossing these mice with raptor deleted animals. So genetic and pharmacologic modulation of the PI3 kinase signaling pathway, specifically mTOR inhibition, can reverse these phenotypes in a very significant way. It also increases survival because of that. So this is the neural stem cell paradigm of congenital hydrocephalus that lends us to round back full circle and think of brain ventricles not only as these, maybe the classic thought of them as these vacuous spaces, right? These passive receptacles, really, that contain brain fluid of this, many mysterious functions, but rather they're windows that, if you use them properly or study them in the right way, they can lend critical insights not only into the pathophysiology of diseases that are relevant for humans and one of the most common congenital brain disorders, but also defining genes that by definition are critical for human brain development. And so what we've really seen and tested in our own patient cohorts is that, usually we think of, congenital hydrocephalus as a disorder really managed exclusively by neurosurgeons. And I think for that reason, it's a historically understudied condition that maybe not a lot of folks know about, in contrast to maybe some of the other neurologic disorders, where in other fields there are clear pathways for neurologists who may spend 80–90% of their time focused on basic investigative work, whereas in neurosurgery, and you'd want it this way for folks who are having their child or themselves operated on, that's not that pathway until recently. Really, it wasn't until about 5 or 7 years ago, where now the NIH has certain platforms set up for neurosurgeon scientists to make contributions specifically in diseases for which they have privileged insight and maybe even patient cohorts that they've developed. And so really the things that we're doing in my own lab are not necessarily novel approaches. I think they're just applied to diseases that are historically understudied. And for that reason, there's a lot of low-hanging fruit. And so we're just not focused on congenital hydrocephalus, but once you set up a platform that has a lot of buy-in, both financially and intellectually and time, then we've been really able to apply it to a few other disorders that we think will not only lend insight into cool aspects of human brain development, but also that are really poorly understood. And so these are just two examples of the paper on below I just brought up because it'll come out next week in JAMA Neurology, where we were able to define the cause of a significant number of temporal lobe epilepsy cases due to mesial temporal sclerosis. We're really excited about that because these are activating mutations, somatic activating mutations in the hippocampus of the RAS signaling pathway. And so it really suggests that we might be able to treat some of these folks with medically intractable epilepsy, and therefore who have to go to the operating room to get their temporal lobe chopped off with anti-cancer agents if it's caught early enough. And because there is a neuroimaging correlate, the MTS, the mesial temporal sclerosis associated with this disease, that could be a real thing. For vein and Galen malformations, this is the most common and severe brain arterial venous malformation of neonates. So if it's untreated, kids die very soon because of arterial blood being shunted, bypassing any developing capillary network directly into veins, and they get massive brain hemorrhages. And so that's a story about RAS1 and FB4 pathway. I'm not going to talk about that, but I'll end on another paper that we had that came out a couple of weeks ago that looks at this other CSF space disease called arachnoid cysts. And I'll wrap up here in a couple of minutes. These are pretty fascinating lesions. Radiographically, they're very obnoxious. They're because they're CSF lesions. They're T2 bright, and they're the most common space occupying lesion in humans in brains. They're really a scourge for neurosurgeons because we really don't know what to do with them if they're not causing obstructive hydrocephalus. It's fascinating because they're completely absent in lower animals that don't have any gyrification or sulcation. And it's probably due to the fact that the arachnoid membrane that layers itself on top of the developing brain really is developing in concert with the underlying brain parenchyma. So this is, again, just another example of a CSF space disorder, extremely common, cared for really exclusively by neurosurgeons, that we think of as a potentially interesting window into brain development and disease. So they're often discovered prenatally during fetal MRI, or in childhood. I talked about before them being treated by neurosurgeons. Again, these kids often have neurodevelopmental disorders, epilepsy, autism. When you get a scan like this is one of my own patients. The obvious point is, how could that not be contributing to these patients' phenotypes? And despite that, when you do a craniotomy or an open cyst fenestration, a lot of times, while it may make the MRI look a little bit better from decompression, it really doesn't change the outcome, neurodevelopmental outcome of this condition. It has been recognized that there are familial forms of arachnoid cyst, and there's increased incidence of arachnoid cyst in several other Mendelian diseases. So that lended the premise and support for investigating this from a genetic point of view. And again, same sort of story where these sporadic cases of arachnoid cysts are, instead of being thought of as just, stuff happens in utero, whether it's due to environment or whatever exposure, is really a genetically driven event due to de novo mutation. And this has, another a gene list. And the fascinating thing about these genes is that they're all highly expressed in neural stem cells, critical regulate epigenetic and transcriptional regulators, a lot of chromatin modifying enzymes. And when you actually look at the expression level of these specific genes involved, they're highly expressed in neural stem cell populations, but also in the developing meninges and arachnoid cell layer, really showing that there is interaction between both the brain parenchyma and its overlying membranes. And through some of the phenotyping, kind of next generation phenotyping methodology that we use, we are beginning to see that, wow, you can actually take these patients with arachnoid cysts and start to subcluster them into different groups. And those different groups are defined by gene mutations, and those different groups have different outcomes, different natural histories. Some are more serious, others are relatively asymptomatic. But sometimes you can define and actually predict the location of where these arachnoid cysts can develop based on the known gene mutation that they have. So again, thinking about exome sequencing as an adjunct for taking care of a patient in a neurosurgery clinic is a bit of a novel phenomenon for us. It's not novel for other fields, right? In cancer or, oncology or hematology. This is a kind of a standard equipment in your toolbox. But for us in neurosurgery, and we've always thought of ourselves as a field of precision. Saltatory leaps within our field have always been tethered to technological innovations that increase precision in what we do. So whether it was the introduction of the operative microscope in the 1970s, or with BrainLab or stealth neuronavigation systems that we use in the operating room to define where we are while we're operating, I would posit that my generation's task would be to increase precision within our field through a molecular approach, right? So being able to define which of these diseases have a genetic basis, how might that influence the choice of different treatments that we employ, whether it's the discovery of a target that can be modulated pharmacologically, or by defining that a patient has a specific mutation, it might tell us, don't do anything, right? That's another kind of obvious concept of first do no harm when you take an oath, the Hippocratic Oath as a physician when you graduate medical school. But sometimes, especially in surgery, not doing anything, especially when we're not clear about how it may actually impact the prognosis of a patient, you can certainly do a lot wrong. Because any surgery can have a complication and a risk, and I can tell you that from firsthand experience. So these are some specific conclusions about the arachnoid cyst study that I'm not going to really go into detail about, but are in that paper, and I can answer any questions, whether after the presentation or directly by email for anybody who's interested. But there's a lot of future directions here, and I think another huge gap in the field that I just want to end with is the ability, or I should say the lack thereof currently, to monitor either CSF production, CSF transit, CSF reabsorption in the brain using non-invasive imaging-based techniques. If we can sort that out, you can apply that to any model. You can apply that to the human patient, and in a non-invasive way. And I think that would be probably, I don't know if it's the holy grail, but that is a very outstanding gap in knowledge, which hopefully, if this talk does anything, it can just get people thinking about doing that. There's a lot of more genes to discover, more mechanisms to elucidate, targets to test in preclinical and clinical translation trials, but it's an exciting time for playing around with some of these rather obscure CSF space diseases. Thank you for your time.